Phonetics should not be confused with phonetics, the study of speech sounds, despite the similarity in pronunciation. In biology, phonetics, PHYNTKs, also known as taxometrics, is an attempt to classify organisms based on overall similarity, usually in morphology or other observable traits, regardless of their phylogeny or evolutionary relation. It is closely related to numerical taxonomy, which is concerned with the use of numerical methods for taxonomic classification. Many people contributed to the development of phonetics, but the most influential were Peter Sneath and Robert R. Sokol. Their books are still primary references for this subdiscipline, although now somewhat dated and out of print. Phonetics has largely been superseded by cladistics for research into evolutionary relationships among species. However, certain phonetic methods, such as neighbor joining, have found their way into phylogenetics as a reasonable approximation of phylogeny when more advanced methods are too computationally expensive. Phonetic techniques include various forms of clustering and ordination. These are sophisticated ways of reducing the variation displayed by organisms to a manageable level. In practice, this means measuring dozens of variables, and then presenting them as 2- or 3-dimensional graphs. Much of the technical challenge in phonetics revolves around balancing the loss of information in such a reduction against the ease of interpreting the resulting graphs. The method can be traced back to 1763 in Adanson because of two shared basic principles: overall similarity and equal weighting, and modern phoneticists are sometimes called neo-Adansonians. So we can distinguish between traditional and numerical phonetics. Difference from cladistics. Phonetic analyses do not distinguish between plesiomorphies, traits that are inherited from an ancestor, and apomorphies, traits that evolved in one or several lineages. Consequently, phonetic analyses are liable to be misled by convergent evolution and adaptive radiation. A typical error occurring in phonetic analysis is that basal evolutionary grades, which retain many plesiomorphies compared to more advanced lineages, appear to be monophyletic. Consider for example songbirds. These can be divided into two groups: Corvida, which retains ancient characters in phenotype and genotype, and Passerida, which has more modern traits. But only the latter are a group of closest relatives. The former are numerous independent and ancient lineages which are about as distantly related to each other as each single one of them is to the Passerida. In a phonetic analysis, the large degree of overall similarity found among the Corvida will make them appear to be monophyletic too. But their shared traits were present in the ancestors of all songbirds already. It is the loss of these ancestral traits rather than the presence that signifies which songbirds are more closely related to each other than to other songbirds. However, the requirement that taxa be monophyletic, rather than paraphyletic as in the case of the Corvida, is itself part of the cladistic view of taxonomy, not necessarily followed to an absolute degree by other schools. The two methodologies need not be mutually exclusive. In general, phonetics is today recognized to provide too much unreliable information about the evolutionary relationships among taxa to remain a mainstay method. But there is no reason why e.g., species identified using phonetics cannot subsequently be subjected to cladistic analysis to determine their evolutionary relationships. Phonetic methods can also be superior to cladistics when only the distinctness of related taxa is important, as the computational requirements are lower. On the other hand, whenever information on the evolutionary history of taxa is needed for a study, a researcher of today will generally try to analyze using cladistic methods. See David Hull's Science as a Process for the History of the Emergence of Phoneticism and Cladism and the Disputes between the Proponents of the Rival Taxonomic Systems. Phonetics today. Traditionally there was a great deal of heated debate between phoneticists and cladists, as both methods were initially proposed to resolve evolutionary relationships. Perhaps the high water mark of phonetics were the DNA hybridization studies by Charles G. Sibley, John E. Ahlquist and Bert L. Monroe, Jr., from which resulted the 1990 Sibley-Ahlquist taxonomy for birds. Highly controversial at its time, some of its findings have been vindicated, while others have been rejected. However, with computers growing increasingly powerful and widespread, more refined cladistic algorithms became available and could put the suggestions of Willy Hennig to the test. As it turned out, the results of cladistic analyses turned out to be superior to those of phonetic methods, at least when it came to resolving phylogenies. Many systematists continue to use phonetic methods, particularly in addressing species-level questions, while a major goal of taxonomy remains describing the tree of life, the evolutionary path connecting all species. In fieldwork, one needs to be able to separate one taxon from another. Classifying diverse groups of closely related organisms that differ by very subtle differences is difficult using a cladistic approach. Phonetics provides numerical tools for examining overall patterns of variation, allowing researchers to identify discrete groups that can be classified as species. Modern applications of phonetics are common in botany, and some examples can be found in most issues of the journal Systematic Botany. Indeed, due to the effects of horizontal gene transfer, polyploid complexes and other peculiarities of plant genomics, phonetic techniques in botany, though less informative altogether, may, in these special cases, be less prone to errors compared with cladistic analysis of DNA sequences. In addition, many of the techniques developed by phonetic taxonomists have been adopted and extended by community ecologists, due to a similar need to deal with large amounts of data.